Hi and welcome. Today we're gonna talk about the WNT signaling pathway. Here's some literature again. So, where does the name come from? The WNT name is an addition of the two words wingless and int. Int1 is a gene which was discovered to induce breast cancer in mice when it had any mutations in it. And the same occurred in the fly Drosophila. There, these mutations led to wingless flies, and the combination of those two words lead to the name WNT pathway. The WNT pathway has different biological functions. The pathway plays a big role in the dorsoventral specification in embryos, so to say the building of back and front. And it plays a role in the control of stem cell niches, in the differentiation of intestinal cells, and osteoblastogenesis. The ligands, which bind to the WNT receptors, are lipoproteins. They have only a small distance signaling role because they're very big and complex, so they can only move on a very small way around the cells. They play an important role in proliferation and differentiation, and those ligands are cysteine rich, and they are palmitoylated, which means they contain a palmitoleic acid compound, which is why these molecules are so big and why they are lipoproteins. We have a receptor which is called the frizzled receptor here. It has a cysteine-rich domain again for the ligand binding, and it has an KTXXV domain for further adapter proteins. They can bind on this domain, and those adapter proteins can, for example, be the disheveled protein. So let's have a word on the disheveled. It binds to the FSD receptor, and it has a region which is called the DIX, the disheveled axon, which binds axon. Axon is a compound of the beta-catenin degrading complex, which we will talk about in a second. And the disheveled destroys the alpha-catenin degrading complex. Here we again see different domains which this protein has. We have the DIX, which is responsible for binding axon, another protein. We have a region which codes for receptor binding sites, and we have another regions which tell this protein where it should be located on the membrane. We had the first receptor, the FSD receptor, and then we have a co-receptor, the LRP5 and 6. The extracellular part contains 4 identical structures called beta propeller, and those beta propellers, they are located here, bind the ligands. In the intracellular domain, we have a region which is called PPS, which can bind the protein axon, and the LRP6 is more important on embryogenesis, and the LRP5 co-receptor has different roles in adult cells. So let's talk about the signaling pathway when it's off. The receptor is not activated. A degradation complex here is built inside the cell, and in this complex we have this beta-catenin. And the beta-catenin is phosphorylized by different kinases here, and this leads to the ubiquination of the molecule. So it's marked for proteasomal degradation, which means the beta-catenin is destroyed by this complex here. It's in the cell, and it's always decomposed. When a ligand binds on our receptor, the receptor is activated, and our co-receptor here is recruited to the FSD receptor, and the receptor here has a binding site for the disheveled protein. FSD binds the disheveled protein, and the co-receptor binds the exin of the degradation complex. The enzyme here stays in place, and the rest of the complex is moved to the receptors, as you can see here. This leads to a conformation change of the complex, and the complex is then destroyed, which you can see here. The proteasomal degradation complex here moves away, and the beta-catenin is no longer destroyed, is no longer ubiquitinated, and therefore it can wander to the nucleus, where it can activate transcription or other cellular processes. I hope this was informational. If you have any questions, just leave them in the comments. I hope you have a wonderful day, and goodbye.